Good everyone. In this video we're going to do biology and we'll cover a topic called DNA replication. So we're doing biology and the topic which we are covering a topic called DNA replication. Okay. Before we can look at the details of this topic, we need first to understand what the central dogma of molecular biology is all about. So, DNA replication, translation, and also transcription, they form what is known as the central dogma of molecular biology. So these are from central dogma of molecular biology. So they are from this branch of or this theory of central dogma of molecular biology. So what is it about the central dogma of molecular biology? So the central dogma of molecular biology is all about a theory, okay? It was a theory which was proposed. So what is it about this same theory? So the central dogma shows what happens for DNA to be converted into another DNA, then from DNA to be converted into RNA, and then this RNA to be converted into proteins. Okay, so what gives the idea of how the conversion takes place is what is known as the central dogma of molecular biology. So what is the process by which DNA is converted into another copy of another DNA? So this process here, the conversion of DNA into another DNA, this is what is known as replication. To replicate is simply to make a copy. So we are simply looking at this branch of or the theory or the part of the central dogma of molecular biology known as replication. Then what is the conversion of DNA to RNA? So the conversion of DNA to RNA is what is known as transcription. Then the conversion of RNA to proteins, this is what is known as translation. So we need to understand these three parts which make up the central dogma of molecular biology, but our interest in this session is based on replication. So let's try to understand something here. We are saying to replicate is to make a copy. Now, what is DNA replication? So DNA replication is simply the process by which a DNA makes a copy of itself. Okay? So DNA make a copy of itself, which is the DNA again. Okay, when DNA makes a copy of itself, which is DNA again, that is known as the replication. And we need to understand that this process takes place in different organelles. Okay, we have two parts of the cell, or two types of the cell rather, known as the eukaryotic or eukaryotic cells, and prokaryotic cells or prokaryotes. So we need to understand that in eukaryotes, DNA replication takes place in the nucleus. But in prokaryotes, because prokaryotes do not have a true nucleus, this DNA replication takes place in the cytosol, or should I say in the cytoplasm. Okay? It takes place in the cytoplasm, but in the eukaryotes, it takes place in the nucleus. Alright, so let's try to understand something here. So when you have a DNA, we know that DNA is a double-stranded. Okay? It's a double-stranded molecule. We can have the 3' end, the 5' end, then we have there the 5' and the 3'. So this is how DNA is. Okay? So this is DNA. It is double-stranded. And its strands are bidirectional. They are bidirectional, meaning that one strand moves from 3' to 5', 3' to 5'. The other strand moves from 5' to 3'. They are bidirectional. Okay? So DNA is made up of double strands. Alright. So let's now understand something here. Okay, so basically about replication, we can have a cell. Okay, this is the original cell, and we have that nucleus. Okay, then this original cell, this is the original. Okay, so when this original cell matures, okay, when it matures, it develops two nucleus. So the nucleus first starts to replicate. It makes a copy of another nucleus. So then this now cell makes a copy of two cells. This is known as replication. So we have the original one, then the nucleus inside divides or makes a copy of itself. Then this nucleus, now there are two in the cell, they now form two cells which are similar. That is known as the replication. Okay, I hope we are able to understand that. So when you have a structure of DNA, like that, okay, with 3' and 5', then here 5', then 3', there is a direction in which this enzyme moves. Okay, this enzyme is known as the helicase. This is the enzyme which is able to separate the two strands. We'll discuss this when we start talking about these enzymes under this same topic. But for now, the point I'm trying to put across here is that in the direction of replication, or in the direction in which this enzyme divides these two strands, that is known as the direction of the replication fork. This is known as direction of the replication fork. The direction in which an enzyme helicase moves. Okay? Direction of replication fork. Okay? So basically that is what you should understand. Okay? And you need to know that the replication fork is rich in AT. Okay? The adenine and thymine. Why is it so? Because the region of adenine and thymine, they are weaker than the region between maybe cytosine and guanine. Why? Because adenine and thymine contain only two hydrogen bonds, but cytosine and guanine contain 3 hydrogen bonds. So it is easy to separate the 2 hydrogen bonds than to separate the 3 hydrogen bonds. Okay, now let's look at the 3 ways which were suggested by different biologists about this same replication. Okay, ways of replication. So there were 3 ways which were suggested. There were what? 3 ways. Okay, let's understand the first way. The first way which was established or which was discovered is known as conservative replication. So the first time it was believed that DNA replication is by conservative replication. Okay, so what is it about that? What is it about the DNA replication by conservative replication? So about this process, it says that when DNA replicates, you have an exact copy and of the original one, and another copy which is just entirely new or fully new. Okay, so when DNA undergoes replication, you have one original copy and a fully new copy. That is what conservative replication is all about. When DNA replicates, you are going to have an exact copy of the original one. So original copy and another one which is just entirely new or fully new. Okay, let me give you an example to understand this. So when you have this DNA strand, so when this DNA undergoes conservative replication, what you are going to have is this. Okay, so allow me to draw this in vertical way, or should I say in vertical way. So I'll draw this in this format. So I'll have this one has undergone replication, so we're going to have an original one, okay, a copy of the original one. Then there will be another copy which will be entirely new. Let me use a different ink. So there will be another copy which is just entirely new. So initially this was the original one. So when it undergoes replication, it has produced this, which is a copy of original, and a copy which is just fully new. We can see that this in green, it's a DNA copy, but it is fully new. But this one is red. In red, it is the DNA which is an exact copy of the original one. As you can see, the original one is in red. This is the original one. The original one is in red. Okay, and the original copy, the copy of the original one, is also in red. And there's also this now, the fully new strand, which is now being formed, which is in green. So basically that is what you need to understand about DNA replication by conservative method. Okay, now let's look at the other method which was proposed. The other method is known as dispersion method. Okay, dispersion method. So what is it about dispersion method? So this method says that when DNA replicates, it will break, okay, at some point. So when DNA undergoes replication, some parts of the DNA will break. Okay, so when you have this DNA, when it undergoes replication, it says that some parts of this DNA will break, and at those points where they break, there will be new segments which are going to be formed. So what it means is that when this DNA undergoes replication, it's at some point it is going to be breaking. Okay, it will break at some point. So this is what I'm drawing here. So it's undergone replication by dispersion, and at some point it has broken. Okay, now at these points which are broken, there will be some segments which are going to be formed like that. Okay, at those points which are broken like that. Okay, like that, like that, like that. Those are the segments which are formed now. Okay, at those points which break. So this method is what is known as dispersion method, and those are the segments. Okay, they are segments which form at those points of breakage. So basically that is what you need to understand about DNA by replication. All right. Now let's look at what is known as the other method which was proposed, and it is called semi-conservative. Semi-conservative replication. Okay, what is it about the semi-conservative replication? So about the semi-conservative replication, it says that when DNA replicates, this is what is going to happen. One, the two strands are going to separate. Okay, the two strands separate, and each of the strands make a different copy. Okay, make a different copy, or makes a copy of a different strand. Make a copy of different strand. So basically that is it about the semi-conservative replication. So let me give an example. For example, if you have this DNA, so once it undergoes replication, so these two strands are going to separate like that, and another strand, these they have separated. Then these now two strands are going to make a copy of a different strand. So each one will make a copy of a different strand. Okay, each one will make a copy of a different strand. Even this one will make a copy of a different strand. So this method is what is basically known as the semi-conservative replication. Okay, so basically that, and we need to understand that semi-conservative method was the best or the correct method by which DNA undergoes replication. So therefore DNA replication occurs by what is known as what? Semi-conservative replication. So basically, that is what you need to understand. Then there was this experiment which was carried out. There's an experiment which was carried out by these two scientists, okay? The Michelson and Stahl. Michelson and Stahl. Okay? This experiment was carried out in 1958 by these two scientists, the Michelson and the Stahl. So these two scientists wanted to prove that DNA indeed occurs by semi-conservative replication. Okay, so they used a certain bacteria. Which is known as the Escherichia. Okay, they used a certain bacteria known as the Escherichia bacteria or the Escherichia coli bacteria. Some call it as E. coli bacteria. Okay, Escherichia coli bacteria. So this was the bacteria which they had to use in order to prove that DNA indeed occurs by semi-conservative replication. So what they did is that they got this E. coli bacteria, so they had to put it in nitrogen 15. So let me draw this. So we have two beakers here and the other one. Okay, so this one contains nitrogen 15, then this one contains nitrogen 14. We know that nitrogen 15 is a heavy nitrogen, nitrogen 14 is light nitrogen. So basically what they did was that they got this bacteria which is E. coli. Okay, they had to put it in nitrogen 15. So they had to put this nitrogen in what is known as nitrogen 15, heavy nitrogen. Okay, so what happened was that so this is the DNA strand. When it was put in this heavy nitrogen, which is nitrogen 15, so this is the DNA strand. When it was put in nitrogen 14, so what happened was this. Okay, so this DNA contained the both strands contained nitrogen 15 because they were put in nitrogen 15 for quite some time. So the two strands contained nitrogen 15. But what happened during the first replication? So during the first replication, okay, so during the first replication, this was what happened. When this DNA has to replicate, okay, they had to separate. One strand, initially these two strands contained nitrogen 15. Even this one contained nitrogen 15. But after they had to split, this was what happened. One strand, okay, let me do this. Alright, so when they had to split these two strands, what's this? Okay. Pardon me for that. So these are the two strands. So these two strands, I mentioned that they were put, this DNA was put in nitrogen 14, which is heavy nitrogen, and then put in nitrogen 14, which is light nitrogen. So during the first replication, this was what happened. One strand, the odd strand, contained nitrogen 14. This one strand contained nitrogen 14 after the first replication. Okay? Then, even here, this strand, we know it was initially put in nitrogen 15. So one strand, the odd strand, contained nitrogen 15. Then the other strand, during the first replication, the other strand contained nitrogen 14. So even here, the odd strand contained nitrogen 15, but the second, during the first replication, the other strand contained nitrogen 14. Okay? Contained nitrogen 14. So this is basically what happened during the first replication. Now, what happened during the second replication? We can see that during the first replication, we had this. We had this again, and went what is known as the second, okay? The second replication. So this was what happened. Okay? So all those strands had to separate. So this was the strand which contained nitrogen 14, even this one, nitrogen 14. Then there was this strand which contained nitrogen 15, okay, and another one which contained nitrogen 15. So this one contained nitrogen 15. Even this one contains nitrogen 15. Okay, so now this one is the one which contains nitrogen 14. Even this one contains nitrogen 14. So during the second replication, this was what happened. So what happened basically was that this strand, again, they had to separate and form new strands. In fact, before we can even talk about what happened during the second replication, let's talk about the ratio. Okay, when these had to form, we can see that there are two nitrogen 14 strands, which are the new strands, and there are how many odd strands? There are two again, odd strand, which are nitrogen 15. If we calculate the ratio or the percentage of the new strand which contain nitrogen 14, we'll discover that since there are two, okay, 2 out of how many strands, the total number is 4, multiplied by 100. This will give us 50%. So 50% were new, meaning that there were new strands which contained nitrogen 14, and 50% were odd strands. Okay, because if you do 2 again divided by the total number of the total strand, which is 4, 2 odd divided by the 4 strands, it will still give us 50% odd. Okay, so we need to know that. Now let's understand something here. Okay, let me just erase this. Okay, let's understand something here. So during the second replication, this was what happened. This was what happened. So during the second replication, this one contained also nitrogen 14, and this one also contained nitrogen 14. This one also contained nitrogen 14, and this one contained nitrogen 14. We can see that there are two pairs of new strands. Then two pairs which contains hybrid. Okay, two pairs contain hybrid, and two pairs contains new. So we can calculate the percentage of new strands from this one. Okay, we can calculate the percentage. We can basically calculate the percentage as follows. So let me just rub maybe the. So this is the second replication. Let's try to calculate the percentage of the new strands. The new strands are the ones which contain nitrogen 14. So how many strands contain nitrogen 14? 1, 2, 3, 4, 5, 6. There are 6. 6 out of. What's the total number of strands? 1, 2, 3, 4, 5, 6, 7, 8. There are 8. Then when you multiply by 100%, what is 6 over 8 multiplied by 100%? So basically this will give you 75%. So 75% were what? New. Okay, let me just rub this part. So 75% were new. So if we subtract 75 from 100 to find the odd strands, we are going to find that the odd strands were basically 25%. Okay, 25% odd. So that is how you can calculate. Alright, so we can say that when this happened, okay, when this happened, they were put in a test tube. They were put in this test tube. They were put in this test tube. So during the first replication, before even replication, the nitrogen 15 was there. Okay, that was nitrogen 15 because it was put in the nitrogen 15. Now what happened during the first replication? So this is now during the first replication. Okay, this arrow pointing down, it is during the first replication. So during the first replication, this line moved to this. Okay, it is here. This strand is here because nitrogen 15 is quite heavy. Okay, but when they had to do now the first replication, it contained nitrogen 14 and nitrogen 15. This one contained nitrogen 14 and nitrogen 15. So it is a bit heavy. Instead of being at that point, it had to shift because it has become a bit light. Okay, then during the third replication, so this was what happened. So there is this one which contains nitrogen 14 / nitrogen 15, and there was entirely this one which contained just nitrogen 14, and it is a light nitrogen. As you can see, the line there, the strand is just a bit far away from this other strand which has a mixture, but this one is just entirely nitrogen 14 and it is light nitrogen. So this is what happened. Now let's look at the steps of DNA replication. Okay, it is very important that we understand the steps of DNA replication. What are the steps of DNA replication? The first step you must understand is this one. Okay, so the first step is known as insertion. So insertion, what does it mean by insertion? This is number one. Okay, so what does this mean by initiation? To initiate is just to start. So initiation basically is just the starting. Okay, the starting of the process of replication. Then what is the second step? The second step is known as the elongation. This step is also known as the polymerization, because when you're dealing with elongation, you are simply forming a chain, or you are forming what? A polymer. No wonder it is called the polymerization. So this one is about forming a long chain known as a polymer. Okay, then let's look at the third step. The third step is what is known as termination. What is it about termination? To terminate something is just to kill or end something. Okay, so to terminate is to end something. So this is just the ending process. Okay, so that's what you need to understand. So after termination, there's another process which takes place. This process, let's say this is number 4, it is known as the modification. Modification is just to make some changes and some corrections. You're just trying to modify something. That is simply modification. Okay, so let's look at the enzymes now. Let's look at the enzymes under replication. So we have this DNA strand. Let's have a DNA strand. Okay, we have this DNA strand like that. Okay, so that is the DNA strand we have. Let me do it. Okay, so this is the DNA strand we have. So the first enzyme which begin to operate is known as the DNA. DNA. So what does this DNA do? So this DNA locates what is known as the origin of replication. This locates what is known as the origin of replication. Okay, so the origin of replication, or where the replication will occur or happen. Okay, so this is what this enzyme DNA do. It just locates the origin of replication, where replication will start. Okay, then there is another enzyme. So this is the DNA strand. There is another enzyme now which begin to separate these two strands. This is the enzyme known as helicase. So enzyme helicase, what it does is that it begins to break these bonds which are between the two strands. Those bonds which are known as phosphodiester bonds. Phosphodiester bond. So this DNA breaks those phosphodiester bond. It begins to break so that to separate the two strands. Okay, so that is the major function of helicase. Now, if you have this DNA like that, let me draw it in this way. Okay, if you have it like that, that is simply the 3' end, the 5' end, the 3' end here, then of course the 5' should be the 5' that one, so 5' end. Okay, then we have this enzyme, the helicase, which has begun to break these two strands into two, to separate the two strands, or to break the phosphodiester bonds. Okay, now there is another enzyme. There's another enzyme as this is breaking. This is helicase. It is breaking. The helicase is breaking what the phosphodiester bond in order to separate the two strands. There's another enzyme now which prevents this from joining again, because you discover that this, as this is being separated again, it might begin to join again. So there's another enzyme which prevents now these two strands from coming together or from joining again. That enzyme is known as the single-stranded binding protein. Single-stranded. The single-stranded binding protein. I call it SSBP. Okay? So that is the enzyme now which prevents these two strands from coming together or from joining. Now you have this DNA. Okay? There is another enzyme which remove. Have you seen that the DNA forms the coils? It is coiled. Now there is another enzyme which remove these coils or the supercoils. Okay, that enzyme is called the topoisomerase. Topoisomerase. This topoisomerase, don't worry, it has the other name. The other name is known as the DNA gyrase. DNA gyrase. Okay, it is also known as the DNA gyrase. So this one removes supercoils so that DNA can just be straight like that. Okay, the two strands can just be straight, so it removes these supercoils, the coils. Okay, so that is what you need to know about the enzyme topoisomerase. Then we have another enzyme. Okay, we have another enzyme. So we have this 3 prime, the 5 prime, then we have the 3 prime there, the 5 prime, then let's say those are the two strands. Okay, now we have this enzyme known as the RNA primase. So the RNA primase, what does the RNA primase do? This RNA primase adds a primer. Okay, a primer is just a nucleotide, so it adds a primer. Usually it adds a primer on the leading strand. So this strand running from 3 prime to 5 prime, that is known as the leading strand. So it adds one primer on the leading strand, and it adds several primers on this other strand known as the lagging strand. So on the lagging strand, it adds several primers. Okay, on the lagging strand, it adds several primers. Lagging strand. This is the lagging strand. So what point am I trying to drive across here? There are two strands. Okay, we have this strand running from 3 prime to 5 prime, then there's another strand which runs from 5 prime to 3 prime there. Okay, so this strand which runs from 3 prime to 5 prime, that is known as the leading strand. Then the strand which runs from 5 prime to 3 prime, that is known as the lagging strand. Lagging behind, right? Remaining behind, following from behind. That is the meaning of lagging. So it is from 5 prime to 3 prime. That is the lagging strand. Okay, now we've said that the RNA primase just adds one primer on the leading strand and adds several primers on the lagging strand. Okay, now let's look at this same DNA here. Let's have this DNA here. Okay, we have this DNA. So, and we have 3 prime there, we have 5 prime there, we have 5 prime there for this one. Okay, then this one is simply the, okay, it is running from 5 prime then going to 3 prime. Okay, so we have that. Okay, this is the 3' end. Now there's another enzyme. There's another enzyme known as the DNA polymerase. We talked about polymerization. So this DNA polymerase continues to form new strands. Okay, let me just wrap this. This is the one now which continues to form new strands of phosphodiester bonds. Forms new strands of phosphodiester bond. This phosphodiester bond. So this is the one now which continues to form phosphodiester bonds. So this run from 5' to 3'. So it begins to form phosphodiester bonds, it begins to form phosphodiester bond, but this run from 5' to 3'. It forms phosphodiester bonds from 3' to what? 5'. Okay? Now, let's understand something here. So, what you need to understand is that this same DNA polymerase 2, okay, it is known as the DNA polymerase 2. So this DNA polymerase 2, it forms strands running from 3' to 5'. It forms strand running from 3' to it forms phosphodiester bonds on the strand running from 3' to 5', and these strands are continuous. So on the 3' to 5', it forms what are known as what? Continuous phosphodiester bond. Continuous phosphodiester bonds. So on the 3' to 5', it forms phosphodiester bond. But we know that, we have understood that this DNA polymerase 2, it can only form the phosphodiester bond in the direction of 5' to 3'. Okay, it can only form this in the direction 5' to what? 3'. That's the only way it can operate, or it can form the phosphodiester bonds. Okay, that's the only way. No way other than that. Then on the 5' to 3', it forms what are known as the Okazaki fragment. Okay, it forms, so DNA polymerase 2, it forms what are known as Okazaki fragments. It forms Okazaki fragments on the 5' to 3' strand. Okay, so we need to understand also that it is very important for us to understand. So 5' to 5', we said that is the lagging strand, right? So it forms Okazaki fragments on the lagging strand. But there is another enzyme which continues to form on this same 5' to 5'. There is another enzyme 5' to 3'. There is another enzyme which is known as the DNA ligase. Please don't confuse this with DNA gyrase. We've talked about DNA gyrase. For gyrase, for DNA gyrase, we said this is also known as the topoisomerase. This one is the one which removes the supercoils. But we have DNA ligase. The DNA ligase, in fact, that is supposed to be C there. DNA gyrase, SE. Okay, the DNA gyrase, this is the one now which continues to form This continues to form phosphodiester bonds. Okay, it continues to form phosphodiester bonds on the lagging strand, the strand running from 5' to 3'. Alright, so let's have an example. Let's say we have this. We have a. Let's say we have on the leading strand. On the leading strand, we have, let's say, 3' A, maybe G, then C, A, G, T, then 5'. This is on the leading strand. Okay, so what are we going to have on the lagging strand? Okay, on the lagging strand, we know that 3' corresponds with 5'. A correspond with T. Okay, adenine correspond with thymine. We have guanine correspond with cytosine. Cytosine correspond with guanine. Cytosine again with guanine. Adenine correspond with thymine. Guanine correspond with cytosine. Thymine correspond with adenine, and 5' correspond with 3'. So this is on the lagging strand. So let's now look at what is known as, we talked about modification, right? We talked about modification as just the correction of the errors. Okay, the correction of errors, modification. So there is this enzyme which we've talked about. We've talked about DNA polymerase. We've talked about DNA polymerase what? We've talked about DNA polymerase 2. Okay? So for DNA polymerase 2, we said that this one, the DNA polymerase 2, is the one which forms the phosphodiester bond. This is the one which forms the phosphodiester bonds. But you need to know that there's also what is known as DNA polymerase 1. Okay? DNA polymerase 1. Just DNA polymerase 1. So, what is it about the DNA polymerase 1? So, this contains what is known as the nuclearase. Okay? It contains the nuclearase. So nuclease is just an enzyme. Enzyme nuclease. Okay? So the function of this DNA polymerase 1 is just to do the correction. It does the correction of errors. Okay? It does correction of errors. That is for what? For DNA polymerase 1. What about for DNA polymerase 2? DNA polymerase 2. What does it do? Oh, polymerase 3. We've talked about DNA polymerase 2 as it forms phosphodiester bonds. Okay? In the 5' to 3' direction. Now, what about the DNA polymerase 3? What does it do? So this one is correct. It also contains enzyme nuclearase, which corrects also what? Errors. But what's the difference between DNA polymerase 1 and DNA polymerase 2? For DNA polymerase 3, DNA polymerase 1, DNA polymerase 3. DNA polymerase 3 only corrects errors in 3' to 5' just. Okay? It only corrects errors in these strands. In the strand running from 3' to 5'. So basically this is what you need to understand about DNA replication. Okay? So in the next part we'll just do the questions on this same topic and will be warm and dry. Okay? Just to cement the concept. Thank you for watching. Have a great day.